get started. So welcome back, everyone. Just a reminder to start with that the final exam is coming up on the horizon. So it's on 1st of December in the morning. The final tutorial class, which starts this week, will be aimed at helping to prepare you for the final exam. So it will include more information on the format of the exam, as well as some practice questions that you can do during class. And I know that for some of you, you won't have that tutorial class until sort of towards the end of next week. And so I've posted some of the slides that will be used in that session on Canvas already that have the sort of general information about the format of the exam, in case you're keen to hear about that earlier. Okay, so this week we're going to focus on memory. And memory is a concept that we all have an intuitive understanding of. So it includes our ability to recall experiences that we've had in the past and to remember facts about the world. We can also think of memory as being quite a broad concept. So anytime something that has happened to us in the past is affecting our current thinking or behavior, we can consider that a type of memory. A very simple example is our experience of food. So there was a time in your life when you had never tried laksa before. Maybe you went to the hawker centre or the food court at the mall. You ordered laksa for the first time. You probably enjoyed it, and then next time you're at that same food court, you decided to order the laksa again. So that very simple kind of example of memory at a biological level implies some interesting things, I think. So one thing that it implies is that there are lasting physiological changes that are happening in our brain in response to the experiences that we have. So in other words, that first time that we ate laksa, something literally changed inside of our brain. Because if nothing changed in our brain, in our nervous system, then there'll be no way for that previous experience of eating laksa to have any effect on our decisions or our behavior in the future. There'll be no way for us to even remember that we had tried laksa before. And so the study of memory in biological psychology is often about trying to understand how the brain is changing in response to our experiences, and in a way that can actually modify our future thoughts and feelings and behavior. So in this lecture, I'm going to start by zooming into the cellular mechanisms of memory. So meaning chemical changes that are happening in the neurons in our brain that allow them to change their function in response to experience. And the idea here is just to give you an initial sense of how it's even possible for our brain to form new memories or new associations. We'll then look at more of the broader picture of what the different types of memory are that we have as humans. And we'll also look at some of the brain areas that are essential to our memory. And in this lecture, I'm going to focus in particular on the hippocampus as a key area of our brain that plays a role in our long-term memory. And then at the end, I'll talk briefly about Alzheimer's disease as a form of memory loss. So by the end of the lecture, you should be able to explain the cellular mechanisms that allow neurons to change with experience, contrast different types of memory, and discuss the function of the hippocampus in memory. So a key way that our brain can change its function with experience is referred to as synaptic plasticity. And this just refers to changes that occur in the function of synapses that exist between neurons in our brain. So one example of this is where a particular synapse might become stronger, so meaning that an action potential that occurs in one neuron, like this neuron here, for example, is more likely to trigger an action potential in another neuron that it's sending signals to. And so you could think of this as a kind of strengthening of the connection between two neurons. Synaptic plasticity is important because it can potentially alter the way that a network of neurons in our brain is processing information, and can also lead to different behaviours being produced by a person or an animal in response to a particular situation or pattern of sensory input. So, for example, when we're eating laksa, it leads to this strengthening of connections between parts of our brain that respond to the smell of laksa and parts of our brain that are involved in producing our experience of like pleasure or reward. Then next time we're exposed to that smell of laksa, it may trigger a kind of anticipation of reward or a craving for laksa that we didn't previously have. Many of the early studies of synaptic plasticity in cognitive neuroscience focused on a particular animal, which is shown here, called aplysia, which is a type of sea slug. And these studies focused on a very simple behaviour that this slug produces, which is called a withdrawal response. And this is where, so on the left here, we have a sort of diagram of the underside of the slug. And the behaviour is where when something touches the belly of the slug, it withdraws its gill deeper into its body. So it's as if it's trying to protect its gill from something that's, touching it. And that very simple behaviour is mediated by a very simple neural circuit in the body of the slug, which is illustrated here. It's where sensory neurons detect touch on the slug's body. They form a synapse with motor neurons that send signals to control the muscle that can cause a withdrawal of the gill into the body. So when something touches the surface of the slug, it can trigger action potentials to occur in the sensory neuron. These can travel down to the synapse and trigger a release of neurotransmitter onto the motor neuron. That might trigger an action potential in the motor neuron, and when that travels down to the synapse that shares with the muscle, that can cause contraction of the muscle and movement of the gill. And there's a very simple kind of learning or memory that the slug displays that involves this withdrawal response called habituation. And this is where the first time you touch the slug, it will tend to withdraw its gill into its body. But then if you keep poking at the slug, keep touching the slug, over time it will tend to respond less and less until it's not responding to that touch anymore. And so you could think of it in terms of the slug learning to ignore this kind of annoying scientist that keeps kind of poking at its belly. And it turns out that very simple kind of learning, that change in behaviour, depending on sort of past experience that the slug has had, is mediated by simple changes occurring at the synapse in this neural circuit. So it's been found that when this habituation process has occurred and the slug is not responding to touch on its body, there are still action potentials that are occurring in the sensory neuron. So the sensory neurons are still registering touch on the slug's body. The muscle and the motor neuron of the slug is also working perfectly fine. So if you insert an electrode to electrically stimulate the motor neuron and produce action potentials in the motor neuron directly, it will still cause a contraction of the muscle and movement of the gill. So it's not just that the slug's muscles have become tired, but rather what's been found is that there's a change in the release of neurotransmitter at the synapse between the sensory neuron and the motor neuron. That as this habituation process occurs, there is less neurotransmitter being released by the sensory neuron onto the motor neuron, and so it's less likely that an action potential will occur in the motor neuron, and so the slug is now not responding to this touch on its body. And so it's a very simple example, but it's kind of a proof of concept, if you like, that synaptic plasticity, a change in the function of a synapse between two neurons in this case, can lead to changes in an animal's behavior that depend on its previous experience, which is a kind of simple example of learning or memory. Another related behavior that these slugs display is called sensitization. And this is where if a intense or even painful touch is applied to the slug, in the future it will tend to respond more strongly in terms of its withdrawal response to even any kind of very mild touch on its body. Okay, so it's as if the slug has learned that there's something harmful in its environment and it needs to be extra careful in quickly withdrawing its gill anytime something brushes against its body. And it turns out that that behavior also depends on changes occurring at the synapse in this neural circuit, where there are other sensory neurons not shown here that register intense or painful touches on the slug's body. These send signals to interneurons that are able to release chemicals onto the synapse, the presynaptic terminals of the sensory neuron, that change the way that this synapse functions in the future. The effect that it has is that next time there's touch on the body of the slug that triggers an action potential in the sensory neuron, that action potential will cause a longer release of neurotransmitter than it did before. And that leads to greater stimulation of the motor neuron and a more intense withdrawal response to even sort of more mild touch on the slug's body. So these studies in slugs were important in illustrating how simple changes in the function of a synapse can lead to changes in behavior. So leading to an animal changing its behavior just in response to the kind of recent experiences that it's had. Okay. In mammals, including humans, there is a particular form of synaptic plasticity that's been studied in detail, and this is called long-term potentiation, or LTP. And this refers to an increase in the strength of a particular synapse that can last for an extended period, so a matter of weeks or even longer. And because of that long-term change in the function of the synapse that occurs in LTP, it's a potential mechanism for how longer-term memory and learning can be achieved in our brain. So longer-term changes in the function of neural networks in our brain. And it's been studied extensively in cells of the hippocampus, which we'll see a bit later in the lecture is a key region of our brain that's been implicated in how we kind of lay down long-term memories, how we form long-term memories. So long-term potentiation is illustrated here. So here we're focusing on a particular neuron labeled A, and this is its dendrite here, and it's receiving signals across synapses that it shares with several other neurons labelled B, C, and D. The plot on the right here illustrates the change in voltage that occurs in neuron A whenever an action potential occurs in neuron B. This first part of the plot here, what it's illustrating is that anytime an action potential occurs in neuron B, it releases neurotransmitter across this synapse onto the dendrite of neuron A, and that causes a small depolarization of neuron A, which is shown here. In the next phase of the experiment that's been illustrated here, there's simultaneous activation of both neuron B and neuron C. So both of these neurons are sending signals at the same time to neuron A. They're both releasing neurotransmitter at the same time onto neuron A. And that leads to spatial summation of those synaptic signals, like what we discussed earlier in the course, where together it leads to a greater depolarization of neuron A, which is illustrated here, a greater change in the voltage of neuron A. And then the interesting part is that following that period, now whenever there is an action potential in neuron B just by itself, when it's sending signals to neuron A just by itself, it still leads to a strong depolarization in neuron A, stronger than it was capable of before. So what this reflects is that that particular synapse between neuron B and neuron A has been strengthened so that whenever there's an action potential in neuron B, it leads to a stronger depolarization in neuron A, increasing the chances that neuron A will also produce an action potential. So LTP relates to this idea proposed by Donald Hebb that a cell that has successfully stimulated another cell in the past will become even more successful at doing that in the future, which is captured with the phrase that neurons that fire together wire together. Another way of putting it is that neural pathways in our brain that are activated may become stronger. And so you're probably aware of all these kind of like brain training apps and things like that. The idea that as we get older, we should use our cognitive functions regularly to sort of keep them strong, and that idea is based off this kind of principle that using functions of our mind, using particular neural pathways may help to activate them and make them stronger in the same way that using our muscles makes them stronger. Long-term potentiation is a process that has been studied in a lot of detail, and I'm going to sort of go through some of the chemical processes that underlie it now. So it's been found that long-term potentiation usually happens at synapses where glutamate is released. So we saw earlier in the course that glutamate is the most common excitatory neurotransmitter that's found in our brain. At a typical excitatory synapse, which is depicted here, we have the axon terminal at the top of the presynaptic neuron, which is releasing the neurotransmitter glutamate into the synaptic cleft. Glutamate is able to bind to ligand-gated receptors in the membrane of the postsynaptic cell, which is illustrated at the bottom here. And for many of those receptors, when glutamate binds to the receptor, it causes an ion channel to open in the receptor that allow positively charged sodium ions to enter the postsynaptic cell, which makes the voltage of the cell more positive, and so depolarizes the cell. It turns out that there are different types of these ligand-gated ion channels that glutamate can bind to, and two of those are labeled here. we have AMPA receptors that function in just the way that I described, in the kind of typical way of a ligand-gated receptor. And then we also have what are called NMDA receptors, illustrated here. And they operate slightly differently. And so a feature of these NMDA receptors is that when the cell is at rest, close to its resting potential, the postsynaptic cell, the NMDA receptors tend to be blocked by the presence of magnesium ions. Okay, so magnesium is another type of ion that's positively charged, and there are many of them floating around in the extracellular fluid, and they happen to be just the right size and shape that they can block the NMDA receptor, but they're not able to pass through the NMDA receptor. Okay, so due to the positive charge of the magnesium ion, it's attracted to the negative charge that's inside the postsynaptic cell when it's at its resting potential. But even if these NMDA receptors have glutamate bound to them, and they should be opening, at rest the magnesium ions will be sitting in the channel and blocking those NMDA receptors so that other ions are not able to enter the cell. And that changes when there's a strong depolarization of the postsynaptic cell. And so we talked earlier in the course about how stronger depolarizations of a cell can occur due to things like temporal summation of synaptic signals or spatial summation of synaptic signals. So in some situations, the presynaptic neuron may repeatedly release neurotransmitter, and that will continue to stimulate the postsynaptic cell and lead to a greater depolarization. So that's temporal summation. And then spatial summation is also illustrated here. It's where the postsynaptic cell may be receiving signals from multiple synapses at once, and together that leads to a stronger depolarization of the postsynaptic cell. So that's also what we were looking at in this example here. And when there's a strong depolarization of the postsynaptic cell, it causes these magnesium ions to be displaced away from the NMDA receptors. And that's because the magnesium ions, being positively charged, are attracted towards the postsynaptic cell when it's at rest and has a negative voltage inside. But when it's more strongly depolarized, it's becoming more positively charged inside, the magnesium ions are no longer attracted, and so they move away from the NMDA receptors. And the significance of that is that when glutamate is released, it can bind to these NMDA receptors, open the channel in the NMDA receptors, and that allows different types of ions to enter the postsynaptic cell. And importantly, including calcium ions that are able to enter through NMDA receptors in particular. And these calcium ions have a special function at the synapse. They're able, once they enter the postsynaptic cell, they're able to bind to other molecules inside of the cell, triggering a series of chemical reactions that leads to changes in the expression of genes inside of the cell that can lead to various physical changes occurring at the synapse, including the increased production of AMPA receptors. Okay, so after calcium has entered the cell due to the opening of the NMDA receptors, it can trigger these changes, including the increased production of AMPA receptors that will be embedded in the postsynaptic membrane of the cell. And that greater density of AMPA receptors in the cell means that in the future, when glutamate is released again, there'll be more receptors that it can bind to, and so it will tend to have a greater effect in depolarizing the postsynaptic cell. Okay, so that type of process, that physical change occurring in the structure of the synapse, can lead to this type of effect where after a strong depolarization of neuron A, in the future, the synapse between neuron B and neuron A will be stronger or more effective, so that the release of neurotransmitters by neuron B will have a greater effect in depolarizing neuron A. So that's long-term potentiation. And it turns out that, so that process is also illustrated here. So before LTP occurs, we have some number of AMPA receptors embedded in the membrane of the postsynaptic neuron at this synapse. After LTP has occurred, we have a greater density of those AMPA receptors that glutamate is able to bind to. And then there are other physical changes that can occur at a synapse as well during LTP. There can be an increased number of vesicles being stored in the axon terminal of the presynaptic neuron, meaning that in the future, when an action potential arrives, there is more neurotransmitter sitting there ready to be released onto the postsynaptic neuron. And that again can lead to a stronger effect of that action potential on the voltage of the postsynaptic neuron. And then there can even be new synapses that are formed during LTP, where previously there may just be one synapse between two neurons. This process of LTP may stimulate the production of more synapses between those two neurons, meaning that next time an action potential arrives, it can have a greater effect on the voltage of the postsynaptic neuron. So the general point here is that this process of LTP, where strong depolarization of a neuron leads to these physical changes in the structure of the synapse, can occur through different mechanisms, but ultimately leads to a strengthening of the connection between those two neurons. Okay, so the key learning points here are that memory requires a physiological change to occur in the brain in response to our experiences. A likely candidate for how this is occurring in our brain is synaptic plasticity. And long-term potentiation is this one example of synaptic plasticity that's been studied in detail, where we know quite a bit about the underlying chemical processes that it involves. And memory and learning may ultimately involve altering the kind of wiring diagram of the brain. So how neurons are connected up together or functioning together as a network through these kind of changes in the strength of synapses occurring in many different places throughout the brain. Okay, so we can do a poll everywhere question at this point. So the question here is to imagine that you take a drug that blocks NMDA receptors in your brain. Which of the following would you predict? Do you think that this drug would both interfere with the formation of new memories and also abolish existing memories that you had before you took the drug? Do you think the drug would interfere with the formation of new memories but not affect the existing memories that you had? Would it abolish existing memories but not affect the formation of new memories, or do you think it would have none of the above effects? Okay, so most people in the class said option B, if you option A or D, and so option B is right, meant to be the correct answer here. So so the idea here is that we've seen that these NMDA receptors at the synapse have this special role in allowing calcium ions to enter the postsynaptic neuron, which are able to trigger these intracellular processes in the neuron that lead to structural changes in the synapse. So the activation of the AMPA receptors, for example, is mainly just having the effect of depolarizing the neuron, whereas the activation of these NMDA receptors is triggering these sort of longer-term changes in the synapse, the strength of the synapse. And those longer-term changes include things like creation of new synapses, the production of more AMPA receptors. And what that means is that next time an action potential happens in the presynaptic neuron, there's more scope for it to have a stronger effect on the postsynaptic neuron. And so if we take a drug that blocks NMDA receptors but doesn't block AMPA receptors, it means that those existing structural changes that happened in the past that affect the function of the synapse will still be there. There'll still be more AMPA receptors, for example, and that will still affect, determine the function of the synapse. But blockage of these NMDA receptors means that it will be more difficult for further structural changes to occur. And so the idea here then is that this type of drug would interfere with the formation of new memories, assuming that that relies on synaptic plasticity, the change in the strength of synapses in our brain, but wouldn't necessarily affect existing memories, assuming that relies on these changes in synapse strength that have happened in the past and are still present there in our brain. Okay, so now we can look a little bit more at the broader picture of memory. I think most of you will be familiar already with the distinction between short-term and long-term memory. And so our short-term memory is our ability to recall information and events that we've just recently experienced, like within the last minute or so, for example. And so if I show you a string of letters on the slide for a few moments and then take them away again, you can still remember what those letters were or what some of them were. So that information is still there in your mind. That's your short-term memory. But if you're, in a minute's time, when your attention has moved on to something else, you may find that you can't remember those letters anymore. That information has kind of gone, has faded away from your short-term memory. Short-term memory is defined partly by that feature that the information that we have in short-term memory tends to quickly fade away. We tend to lose that information if we're not actively paying attention to it or kind of rehearsing it in our mind. And it's also defined by a limited capacity. There's only so much information that we can hold in mind at once. Our long-term memory refers to our ability to recall events that have happened in the past. And the past might mean 10 minutes ago, yesterday, last week, or 20 years ago. A long-term memory has a very large capacity. And so one example of that is our memory that we rely on for language, that we all know many, many thousands of words. And so that's a lot of information that is stored in our brain throughout our life. Our long-term memory doesn't necessarily fade away when we're not paying attention to it. And so, for example, if I asked you to write down the names of your classmates when you were in the first year of primary school, that's probably a piece of information that you haven't thought about for some time, but you might still be able to recall some of your friends' names in primary school. And if you went home and looked up some photos of your class, that seeing their faces might then jog your memory and allow you to recall even more of their names. And so the point being that information is in there, in your brain somewhere, even if you're not thinking about it for many, many years, and even if you don't always have complete access to it. So the idea here is that all the information that we're conscious of is initially entering our short-term memory, but then only a fraction of that is being consolidated into a longer-term, more stable form of storage. And then there are different types of long-term memory that we can define as well. One distinction is between explicit memory, which refers to memories that we're consciously aware of, and implicit memory, which refers to effects of our past experience that can, on our current thinking and behaviour, that we're not necessarily aware of, that are happening at more of an unconscious level. As an example of implicit memory, one example is a process called priming. And this is where exposure to one kind of stimulus, so something that we see or we hear, can then influence our response to another stimulus. So for example, if I prime you in a particular way by showing you different images from the bathroom, and then I ask you to complete the word here, so fill in the missing letter to make a word. You might find that, for many of you, that the first word that you think of is the word soap, whereas if I had primed you with pictures of food and the like, the kitchen, then maybe you would have thought of the word soup instead. And so that may or may not have worked, but the idea here is that previous experiences, things that we've recently been exposed to, can sometimes influence our thinking and behaviour in ways that we're not always sort of fully aware of. You can imagine this type of priming process may be happening to an extent in day-to-day life, where things that we see are kind of influencing our thinking without us necessarily being conscious of that happening. Another example of implicit memory is what's called procedural memory, which relates to our ability to learn new motor skills and kind of everyday tasks and habits. And so we've all had the experience of learning like a new sport, for example, like tennis, learning how to swim, how to drive, how to ride a bike. And when we learn these kind of motor skills, often at first we can be, not very good at it, very clumsy and uncoordinated when we're playing tennis for the first time ever. But then just with a bit of repeated practice, we quickly get a lot better at it. And oftentimes we're not necessarily aware of how we're getting better. We know we're practicing, and maybe we can read the flight of the ball better, we can perform a particular pattern of coordinated movements better, and that's leading us to hit the ball more accurately. But a part of that learning is more like kind of muscle memory. It's a kind of learning from experience that is happening at a more of an unconscious level that we can't necessarily describe to another person. Okay, and so explicit memory is often also referred to as declarative memory, and this refers to information that we can deliberately, bring to mind and explain to someone else or describe to someone else. There are two main types of declarative memories. One is episodic memory, which refers to our memory for personal events and experiences that we've had in our life. And so if I asked you who you had lunch with yesterday, you would probably answer that question by bringing to mind a particular experience, a particular moment in your life. You might be able to picture what you were eating, where you were, who was there, where they were sitting around the table, what you talked about, and so on. These kind of autobiographical details of that experience. In contrast, semantic memory refers to more abstract information, facts and concepts that we're aware of that are not linked to any particular moment in our life. And so if I ask you who was the first Prime Minister of Singapore, you can probably answer that question without thinking of any particular moment in your life. So these kind of definitions I've gone through are not always adequate for describing every feature of our memory. You may be able to think of kind of grey areas, like examples of memory that don't fit neatly into one of these categories. But they're still kind of useful distinctions to make. And one reason for that is because they're helpful for if we're trying to identify the biological processes that underlie our memory. Because if we know that some of our memories are long-term, they form and they remain and we have access to them for 20 years, for example, then that implies that if we're looking for the biological basis of long-term memory, we need to be looking for a change that's happening in our brain in response to experience that persists there for many, many decades, right? In contrast, if we know that short-term memory tends to only last sort of for a minute or so and then disappears if we're not sort of actively rehearsing that information, then it means to find the biological basis of short-term memory, we need to be looking for processes happening in our brain that are persisting for a minute or so and then kind of stopping, if you like. The textbook uses this kind of arcane term, an engram, which is sometimes used in the literature to refer to a physical representation of something that's been learnt. And so if our long-term memories require changes in the strength of synapses in our brain, for example, then those changes in the strength of synapses would be the engram of those memories. And so these engrams are likely to be different for different types of memory that we have. Okay, so the key learning points here are that short and long-term memory can be distinguished in terms of their capacity and their reliance on attention for continued storage. And then we can also distinguish between these different forms of long-term memory. So episodic and semantic memory and procedural memory, for example. Okay, so one more poll everywhere question. So what is your name? So to answer this question, what type of memory are you using? Semantic, episodic, or procedural memory, or some mixture of the above? Okay, so so most people put A, some said D as well. And so, I agree the most obvious answer and here is semantic memory, that our name is this kind of abstract piece of information that we have that isn't necessarily tied to any particular kind of moment in our life. I think technically like D is like a decent answer as well. For example, because to answer this question, if someone asks what our name is and we answer the question, we might do that by talking, and speech is reliant on a kind of procedural memory. It's a action, a way of using our body that we've learned. It's a skill that we've learned during life. And so technically you could say that that this relies on sort of a mixture of procedural and semantic memory, for example. But here the idea is that this type of information is really a semantic form of information. Okay. All right. So let's have a break and come back at 5 minutes to 3. Thanks. Okay. So I better get started again. So we've been talking about these distinctions between types of memory, so long-term, short-term memory, and so on. And one of the ways that we've learnt about these different components of memory that we have is by studying the experiences of people who have lost certain components of their memory following damage to their brain. And so amnesia is a term that refers to memory loss, and it's something that can occur following localized damage to the brain. One of the most well-known cases of amnesia in cognitive neuroscience is the case of Henry Molaison, who's also referred to as patient HM in the literature. He was an American man who suffered a high frequency of epileptic seizures that didn't respond well to medication. And because of that, in the 1950s, he underwent a kind of radical surgery that was performed by William Scoville, who's a surgeon, pictured on the right here, who experimented with lobotomy in people with a mental illness. And so where lobotomy is where a part of the brain is intentionally destroyed or cut out. What the surgeon did is remove most of the hippocampus as well as some nearby structures of the temporal cortex in both hemispheres of Henry's brain. And this did apparently have the effect of reducing the incidence of his seizures, but it also caused these profound impairments in his memory that lasted from his surgery until for the rest of his life. So the hippocampus is this structure that's buried deep in the temporal lobe of our brain. It's highlighted in green here. On the right is a photograph of HM's brain taken after he died. So here we're looking at the ventral surface of the brain, and highlighted with the white boxes here is a region of the temporal cortex where the hippocampus is located that is missing in HM's brain, that was cut out. At the bottom, we have structural MRI scans of HM's brain when he was still alive. Here we're looking at a coronal view of the brain, and these dark areas are empty space in the temporal lobes where the hippocampus ought to be. The effect of losing this part of his brain, having the entire hippocampus cut out of his brain, was fairly specific. And so his intelligence still seemed to remain intact. He could still have an intelligent conversation with another person. Apparently his personality was similar after the surgery than it was before the surgery. He still came across as the same person, but he experienced these extreme impairments to his memory. One was an inability to form new memories, new long-term memories, which is referred to as anterior grade amnesia. Another effect was the loss of long-term memories that for things that had happened to him in the years prior to his surgery. Okay, and so this is referred to as retrograde amnesia. And so he was 27 when he had this surgery, and he died many years later in 2008 at the age of 82. And so he lived the majority of his life in this kind of unusual state of being unable to form a kind of lasting impression of things that happened to him. This is a book that was published a few years ago that I found quite interesting, and you might be interested in checking out if you're interested in learning more about HM's story. The author of the book is actually the grandson of the surgeon who performed the surgery on HM, which is a kind of a strange connection. And in this book, he talks about HM's life, the scientists who kind of made a career out of studying him, and also this very dark history of medicine and psychiatry, where in places like the United States around the sort of mid-1900s, these surgeries, lobotomies, where parts of the brain are removed, were performed in thousands of people who had mental illness, often without the surgeon really having much of an idea of what it would do to the person. So to talk a bit more about these memory impairments that HM experienced, I mentioned this anterior grade amnesia, this inability to form new long-term memories. What that meant is that you could go to HM's house, introduce yourself, have a conversation with him, and then if you left the room for 5 minutes and came back again, he probably wouldn't remember having ever met you. because of that, he could have the same conversation again and again with the same person without remembering having done that before. He could also read the same book or magazine again and again without realizing. He had difficulty remembering his own age and what year it was. And apparently he watched the news on TV every night, but he couldn't remember most of what had happened in the world since the date of his surgery. His episodic memory was particularly impaired, so he couldn't describe any personal experience that he had had since the surgery. In contrast, his semantic memories were still, he could form some semantic memories to some extent. So apparently he learnt the names of famous figures like the US president or something like that he saw on TV each night. But his semantic memories were also not normal. He also had difficulty learning certain types of information. So apparently he didn't learn certain kind of words that entered the English language after the date of his surgery. Apparently that includes words like jacuzzi and granola. And then in addition to these problems with forming new long-term memories, whether episodic or to some extent semantic as well, he also had this retrograde amnesia, this loss of memories that he had formed before the surgery happened, before his hippocampus was cut out. This was particularly the case for events that occurred in years recent to the surgery. So he still had memories of information that he had learnt as a child, for example, but he didn't have much memory of things that had happened in the few years just before the surgery happened. And again, this was more severe for episodic memories than semantic memories. So he could only remember a small number of, he only had a small number of clear memories for personal events that had happened to him during his childhood before the surgery. But in contrast, he still had his language. There was still a lot of information that he had learnt as a child that was still there in his brain. He had intact short-term memory. So you could tell him like a phone number, for example, ask him to remember it, and as long as he had the opportunity to kind of rehearse it in his mind, then that information would stay there. But then if he was distracted, or after a minute or so, if he wasn't actively trying to rehearse that information, then it would be gone. So this is a hallmark of kind of normal short-term memory. He also seemed to have better implicit memory than explicit memory, including intact procedural memory. And so procedural memory, as we saw, involves learning new skills, for example. One example that was studied is the ability to read sentences backwards. And so this is a sentence that's been flipped backwards, and you probably find it difficult to read. Apparently, we can get a lot better at this just by practicing it, which is a kind of procedural memory. And the researchers working with HM asked him to do this, to practice reading sentences backwards for like an hour a day, for example. And he ended up getting kind of better at it, even though he couldn't actually remember having kind of practiced this skill. And so this effect of removing the hippocampus from the brain seemed to lead to problems with certain types of memory, but not others. Okay, so particularly with declarative memories, and especially for episodic memories. While short-term memory and other forms of long-term memory, like procedural memory, seem to be relatively intact. This kind of pattern of memory loss, of intact short-term memory, but problems with forming new memories, new long-term memories, has also been observed in other cases as well, following damage to the hippocampus. Another patient that's been studied in detail is this man on the right here, known as patient EP, who suffered damage to his hippocampus following a virus. And I've put a link here to a paper that summarizes some of the research done together with him if you're interested. And his memory problems are illustrated in this video.
No difficulty repeating back a list of words. In this case, dog, raccoon, lion, and parrot.
Dog, raccoon, lion, and a parrot.
I read you 4 animals.
Okay, so here, the researcher has read him a list of words, asked him to remember it, and then she's going to ask him what the words were again, sort of like 15 minutes later or so.
And I want you to tell me yes if you think the animal was on the list I read you about 15 minutes ago, or no if it was not on the list.
Wonderful.
Okay? Cat.
Yes.
How sure are you?
I feel reasonably sure
Reasonably
on that cat.
sure? Uh-huh. Raccoon.
No. No. I feel no.
Giraffe.
Yes.
How sure are you that I read
I'm
you?
real sure on that.
Real sure?
They're beautiful animals,
Okay.
believe me.
Lion.
No.
Okay.
I'm sorry, I forget her name. What's her name?
Her name?
Yeah.
I don't remember.
Okay. Jennifer.
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 Yeah.
I'm sorry.
Have you met Jennifer before today?
I think so. Yes. I'm not positive though.
Yeah.
That's okay.
I'm sorry, my
No.
Oh, that's
It's
okay.
okay.
Yeah.
We all have.
Well, we all get that way.
Oh, sure. How much have you worked with him?
We have probably visited his house 200 times.
200 times. And for him, is each time the first time for him?
Well, you see, it's yes and no. It's clear that he does have a record of the experiences that are based on the habits, the interactions, the emotional values of the interactions, the fact that they've been positive and not negative. He certainly lets us in the door faster.
I mean, like you saw, he couldn't tell you my name.
Yeah.
He couldn't tell you if you saw me before. But there's definitely a rapport there that I think has been built, and he's aware on some level.
Okay, so what do these kind of cases tell us about the biological basis of memory? So one thing they illustrate is how different components of our memory can operate somewhat independently of each other and rely on different areas of our brain. That this kind of radical change in a person's brain of cutting out their entire hippocampus can cause them to lose certain capabilities for memory while still having other capabilities intact, like their short-term memory. And in particular, the hippocampus seems to be implicated in declarative memory, especially the formation of new episodic memories, but less so in short-term or procedural memory. So the fact that removing this part of the brain, in people that have had this part of the brain removed, they still have a short-term memory. They can still learn kind of procedural skills. Is pretty powerful evidence that the hippocampus is not necessary for those features of memory. There's a question of what the hippocampus is doing exactly to enable us to form new kind of long-term episodic memories of experiences that we've had. For example, one possibility is that it's a site where these memories are stored in our brain. Another possibility is that those episodic memories are actually stored somewhere else, but the hippocampus is kind of like a hub, if you like, a structure that's able to withdraw those memories from long-term storage back into the forefront of our mind. And so there's ongoing research into this kind of question. Generally, it's thought that long-term memory relies on an exchange of information between the hippocampus and other areas of the cortex, where the kind of more episodic personal details of events are likely to be stored more so in the hippocampus, while the more semantic information is stored elsewhere in the cortex. The hippocampus has also been implicated in another kind of long-term memory that is quite interesting, I think, which is what's called our spatial memory. And so this is the kind of memory that we use to navigate. So it's our memory for the sort of spatial layout of environments that we spend our time in every day. And so when you came to the lecture theatre from the MRT station or wherever, you were relying on your spatial memory, your memory of the layout of the campus to get yourself here. This spatial memory has been studied partly in rats using kind of experimental paradigm illustrated here called the Morris water maze. This is where you have this tub of water, and somewhere in the tub of water is a platform that's just sort of below the water level, so it can't be seen from the surface. But if you're a rat and you're in the tank and you find this platform, it's high enough that you can stand on the platform, and then you don't have to keep paddling to keep afloat. And so generally, rats sort of put into this tank will swim around. Eventually, they'll find the platform, and then they'll kind of more happily sit on the platform. In these kind of studies that use this type of paradigm, what they do is they study the process of learning, where the rat is able to learn the location of the platform across multiple trials. And so typically they put sort of visual landmarks around the tank, so that once the rat has learnt where the platform is in the tank, in the future, if it's put into kind of a random location in the tank, it can use those visual landmarks to navigate to where it knows where the platform is located. So this is involving a kind of spatial memory for where the platform is located within this tank. Rats typically, learn this over repeated trials. They get faster and faster at finding their way to the platform, but damage to the hippocampus, rats that have had their hippocampus removed, for example, tend to have difficulty with this kind of navigation task. So, for example, rats that have been trained, have learnt a particular, learnt the location of a platform. If their hippocampus is removed and then they're put back into the tank, they seem to have lost that memory of where the platform is located. The hippocampus also seems to be involved in spatial memory in humans as well. And one interesting study that illustrated this examined spatial memory in cab drivers. And these were cab drivers in London. So these are people that have a vast spatial memory of the road network throughout the city that they live in. So this study was done in 2000, which was before Google Maps and the like were commonplace, and so these cab drivers were particularly reliant on their memory. And what they did, first of all, is they invited a bunch of cab drivers into the lab. They had them they scanned their brain, so using PET to track activation of the brain while they were recalling particular routes through the city. So they would ask them, like, what's the fastest route between Heathrow Airport and the Tower of London, or something like that. And as they're kind of thinking of that information, so bringing to mind the kind of spatial layout of the city, they found that their hippocampus became active. And interestingly, they also found that there were structural differences in cab drivers in the hippocampus compared to other people. That these cab drivers with a particularly extensive spatial memory tended to have some parts of their hippocampus were expanded, so larger, and that was particularly the case for cab drivers that had worked in that profession for a longer period of time. Okay, so our ability to remember the layout of environments that we're in seems to rely partly on the hippocampus, and it's been found that there are particular cells in the hippocampus that are maintaining information about where we are currently located in the environment that we're in, and these are referred to as place cells, and they're illustrated in this video. So what you're looking at here is a kind of bird's eye view onto a box. The walls of the box have these different sort of patterns painted on them. These are facing inwards. And here, this is a representation of a rat that was placed into this box during the experiment. And during this particular trial of the experiment, the researchers are using an electrode to record the activity of a single place cell in the hippocampus of the rat's brain while it's kind of walking around this environment. Okay, so this crackling sound is illustrating when there's action potentials in this particular cell that they're recording from as the rat is walking around this box. Okay, so the idea here is they've identified this cell in the rat's brain that is becoming active when it's in a specific location of this environment that it's in. This is illustrated here as well. So this is, again, illustrating sort of a rat walking around a particular environment and a cell in its brain becoming active when it's in a specific location of that environment. And the idea is that we have, both rats and humans as well, have many of these place cells, where different cells will be becoming active when we are in different places of an environment that we're familiar with. So right now you're in the lecture theatre, you have a sense of your location within the lecture theatre, and at the end of the lecture, when you get up and walk towards the door, your sense of location will be changing, and the idea that that information is being represented in your brain in terms of which place cells are becoming more active in your hippocampus. Okay. So I focus mainly on episodic long-term memories and a bit on kind of spatial memory here and the role of the hippocampus in those components of memory. The other types of memory, like semantic memory, implicit memories, and short-term memory, have tended to be associated with other areas of the brain, like the prefrontal cortex, for example, was thought to be important for our short-term memories. I'm not going to talk about these kind of other areas and other types of memory, but if you're interested in kind of learning more, chapter 12 of the textbook goes into a bit more detail about these. So the key points here are that the hippocampus is a structure in our brain that's necessary for encoding new episodic memories and retrieving stored episodic memories. The hippocampus also seems to underlie our spatial memory, our kind of long-term memory of the layout of environments that we're familiar with, while other forms of long-term memory, including procedural memory, seem to be mediated elsewhere in our brain. Okay, so to finish up with, we can talk briefly about Alzheimer's disease. And so Alzheimer's is the most common form of severe amnesia or memory loss. So we've been talking about these kind of unusual cases of people who have lost their parts of their long-term memory following specific kind of damage to the hippocampus, like due to misguided surgeries or a virus. Alzheimer's, on the other hand, is much more common. It usually occurs later in life, so after the age of 60 or so. The textbook estimates that something like a half of people over 85 have Alzheimer's. That's quite a high estimate, and I've seen sort of lower estimates elsewhere. The problems with memory that Alzheimer's includes or involves can include problems storing new memories. So that might mean forgetting that you've had a particular conversation with someone before, forgetting what you did that week, forgetting the names of people you know or objects, and also a kind of disorientation in time and place. And so, for example, tending to get lost when you leave the house because your spatial memory isn't working as it used to. And so these issues with memory are also to some extent a part of normal aging. So all of us will have these kind of problems to some extent, and that can make it difficult to distinguish Alzheimer's from kind of normal aging in the early stages of the disease. But in Alzheimer's, these problems tend to progress to more extreme memory loss, as well as progressing to other kinds of symptoms like a sense of confusion, even delusional thinking, and a sense of apathy and depression. In Alzheimer's disease, there is this progressive loss of neurons in the brain across the cortex. This is referred to as atrophy, where parts of the brain are kind of losing cells and degenerating, if you like. It's illustrated on the right here, where we have a cross-section of a post-mortem brain of somebody without Alzheimer's disease at the top and somebody with Alzheimer's at the bottom. You can see that the brain looks a bit more kind of like shrunken in parts. There are areas where the grey matter is a lot more has a lot more volume in the more healthy brain compared to the brain with Alzheimer's disease, and this sort of expansion of the ventricles, creating these sort of larger holes in the brain, is coming about because of that loss of grey matter around the ventricles. You can see this atrophy of the grey matter in MRI scans of the brain as well. And so again, on the right here, we have a healthy control. On the left, we have a patient with Alzheimer's disease. And you can see that areas of grey matter have sort of lost their volume in the brain of a person with Alzheimer's. Highlighted in red here is the location of the hippocampus. And again, you can see this sort of shrinking of the hippocampus that reflects the loss of cells, the loss of neurons in that area. And so this loss of neurons in the hippocampus and perhaps other areas of the brain as well seems to be what contributes to these difficulties with memory in Alzheimer's. This illustrates the degeneration of neurons that occurs in this condition. So on the left here is a diagram of what a typical neuron from the prefrontal cortex looks like in somebody without Alzheimer's disease. On the right are some diagrams of the kind of changes in structure of the neurons that can be observed in the same area of the brain in people without with Alzheimer's disease. So you can see that there are this sort of progressive degeneration of the structure of the neurons. And so there's a lot of research into what is causing this loss of neurons in the brain. And it's been found that there are particular genes that are linked to Alzheimer's disease. It's partly a heritable condition. And a key pathological feature that's been found to occur are the presence of these kind of odd structures referred to as plaques and tangles in the brain of people with Alzheimer's. And so these are subcellular structures that can be found in neurons and seen under a microscope. And they relate to these particular proteins called amyloid and tau, which are normal proteins that are found in all of our brains. But in Alzheimer's, these proteins tend to kind of clump together and form into these plaque and tangle structures. And the presence of these structures seems to disrupt the kind of usual function of neurons and leads them to die. And so there's research now in trying to understand or trying to find a way to develop drugs, for example, to intervene in this process and prevent these structures from damaging the neurons so that we can halt the progression of the disease. Okay, so the key learning points here are that Alzheimer's is a common cause of amnesia that occurs late in life, or can occur late in life. It's related to the loss of neurons in the hippocampus and across different areas of the cortex. Okay, so we can finish up with a couple more poll everywhere questions. So these questions relate to patient HM. First question is, Which of the following best describes the implications of HM's case for our understanding of how memory is localized in the brain? Does it tell us that human memory depends primarily on the hippocampus, that memory functions are intricately woven throughout the brain with no single location of the brain being especially important for a certain type of memory? Does it tell us that distinct types of memory engage specialized brain structures, each contributing uniquely to our overall memory system? Or does it tell us that short-term memory operates independently of brain structure? Okay, so so pretty much everyone put option C, which is meant to be the right answer. That's great. So it's really kind of this pattern of memory loss where losing this big chunk of the brain can severely impair a particular component of memory, like the ability to form memories for personal events that happen to you, while leaving other types of memory intact. Illustrates this association between different types of memories and specific parts of our brain. Okay. Then one more question. So what insights can we gain from HM's case regarding the role of the hippocampus in episodic memory? You say that the hippocampus is the sole structure responsible for storing all episodic memories. That once episodic memories are formed, the hippocampus has little to no role in their recall. Would you say that the hippocampus is essential for encoding new experiences into long-term memory, acting as a gateway for episodic memory formation? That the hippocampus may be a key storage site in the brain for new episodic memories? Or do you think both C and D are highlighting the likely role of the hippocampus in the memory, in our memory? Okay, so everyone is saying E, which is right. So that's great. So we saw these details of HM's memory loss, that he did have some episodic memories from early in childhood. So the hippocampus can't be the only structure in our brain that's involved in storing episodic memories. But having said that, he did lose episodic memories from years just before the surgery. So this region does have a role in the recall of those memories. And so C and D are both right. But it seems to be essential for encoding new episodic experiences into our long-term memory, and likely acts as a key storage site in the brain for newly formed episodic memories. Okay. Right, so that's all for today. So next week we'll be changing topics a bit and talking about the process of sleep and dreams. So thank you. I'll see you next week.